Okay, now we're going to look at alternative splicing of RNA. And so the idea here is if we have our RNA with the introns still intact, depending upon what cell this RNA is being transcribed in, will determine how it's going to be spliced. So it's possible that in some cells, the exon number 4 gets spliced out along with the adjacent introns, so that you end up with an mRNA that looks like exon 1, 2, 3, and 5. And in some other cell types, this same RNA that has been transcribed from the exact same gene gets spliced in such a way where exon 3 is spliced out along with its adjacent introns. And so what you end up with is an mRNA that looks like exon 1, 2, skip 3, 4, and 5. So the protein product from this messenger RNA up here and this messenger RNA down here are going to give rise to unique proteins. Now, of course, they will have a lot in common, but they are going to be different. And it turns out that 94% of human genes undergo alternative splicing. So although we may have about 30,000 or so genes within the human genome, we're going to have way more proteins in our proteome, simply because of alternative splicing. So our question really should be now, what is the difference between a cell that splices the mRNA like this and an mRNA like that? How is it that they splice that same messenger RNA in two different ways? And the answer is splice site enhancers and splice site silencers. These are sequences within the DNA that are then, of course, transcribed into the RNA, and they're sequences to which specific proteins bind. Let's take a look at splice site silencers first. So whether that silencer occurs within an intron or whether it occurs within an exon, we call it either an exon splice silencer or an intron splice site silencer. Same thing with the enhancers. If that enhancer is with an exon, it's called an exon splice site enhancer, or in green here, an intron splice site enhancer. Now, splice site silencers. Those are going to be in red here, and those are bound by proteins called hnRNPs, heterogeneous nuclear ribonucleoproteins. And these are proteins that bind to these exon splice site silencers or intron splice site silencers. And what do? They silence splice sites. They inhibit the various components of the spliceosome from binding to either the branch site or binding to the 5' splice site. And when you inhibit the proteins from binding to these sites here, then instead of this intron being spliced out and this intron being spliced out, what happens is you end up with this intron, this exon, and this intron all being spliced out, right? Because the U1 bound here cannot interact with the U2 bound here because the U2 here can't bind to the branch site. And because of that, instead of getting the two introns spliced out separately and leaving an exon in, they are all spliced out together, as shown by these sort of light blue arrows here. Furthermore, there are sequences that are splice site enhancers, and there are proteins that bind to those enhancers and help the spliceosome recognize and bind to the introns and exons. So these SR proteins bind to these splice site enhancers and then recruit U1 to bind to the 5' splice site. They recruit U2 to bind to the branch point. So they're going to make sure that each one of those introns is spliced out and that the exon is going to stay within the messenger RNA. Now notice, and this is important, in that these exon splice site enhancers and these exon splice site silencers are, as the name suggests, located within exons, located in potentially protein-encoded regions, protein coding regions of that messenger RNA. And to these sequences are attracted these either SR proteins or these hnRNP proteins. So that means that now, even if we have a mutation that is a silent mutation, a mutation which, like maybe in this little orange spot, that when that mutation occurs, it doesn't change the amino acid that is encoded by it. So it doesn't change the structure of the protein. However, even though it's a silent mutation, it still is a mutation. We're still changing one base to another. And that might be enough structural difference such that these SR proteins or these hnRNP proteins will no longer bind. And if they no longer bind, even though that doesn't change the amino acid sequence of the protein, it might change the splicing of that messenger RNA, which will even have a more drastic effect on the protein itself. And there is a disease out there called spinal muscular atrophy, in which if the people and whoever inherits this mutation, instead of having sort of normal healthy motor neurons, the motor neurons begin to degrade, and the muscles that they are attached to begin to atrophy. And so you're not able, your muscles aren't as strong, and you can't walk, and you have problems with movement. Well, here's the thing. What was discovered is that so here is the codons within the gene called Survival of Motor Neuron 2. So here are the codons, GGU, UUC, AGA, blah, 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 so on and so forth. Well, there's a mutation that they discovered where a C is mutated to a U. That means that the codon here goes from a UUC to a UUU. But look at this. UUC encodes a phenylalanine. UUU encodes a phenylalanine. So they're thinking, the researcher, they go, well, surely this mutation can't be the reason for this disease because it's a silent mutation. Well, what they discovered is that what's happening is that this mutation is causing it is either it is eliminating an exon splice enhancer such that the protein can no longer bind it, or it is creating accidentally a new exon splice site silencer that allows the protein, a silencer protein, to bind to it. Either one of these two models causes this problem, causes these splice sites to be ignored such that what happens is this entire exon is spliced out. This entire exon is spliced out regardless of which model we're looking at. So this is an example of a silent mutation causing a splice defect, a splicing defect, where an entire exon is now missing from the messenger RNA and therefore that portion of the protein. Now, let's talk a little bit about why introns might be selected for, right? We said that the majority of a gene is actually intronic sequence, and yet introns, for the most part, don't encode anything. They don't do anything. So why have they been selected for? Why haven't they been dropped out of the human genome? Well, there's two things you have to understand. First is that exons themselves, individual exons, often encode entirely functional domains of the protein. So maybe this exon here encodes a DNA binding domain, and maybe this exon here encodes an activation domain. And when I say an independently sort of functional part of a protein, meaning if we took this domain here and we attached it onto another protein, that would give that protein the ability now to bind DNA. So it's very modular in that respect. Now, the introns that separate them mean that if a mutation occurs in which there's crossing over between two genes, as long as that crossing over occurs between introns, then it's easy to conceive an idea of where, even though we've got this random mutation, it ends up generating a new functional protein. Whereas if there weren't the introns, and if the introns weren't the largest part of a gene, then most of those crossing over events would occur in an exon and it would interrupt these regions that encode independent domains. So what these introns do is they sort of make genes more modular. They make mutations more likely to generate functional proteins, like these big sort of crossing over mutations. And we can see evidence of that. We can see genes right here, this LDL receptor, we can look at them and we can look at the exons and go, oh, look at this. All of these exons came from this EGF precursor gene. All of these exons over here came from this C9 complement gene. And they came about, probably this new LDL receptor gene then came about from these large crossing over mutations, these recombinant mutations in which two genes got linked together. And because that more likely will occur at a region of an intron, then they remain functional and their exons remain encoding functional aspects of a protein. Right, so in the next lecture, we're going to talk again about the processing of RNA, but in this case, we're going to, instead of looking at messenger RNAs, we're going to look at regulatory RNAs.